Hello everyone, welcome you all in the another video lecture. So in this video tutorial, we will discuss about Ziehl-Neelsen staining method. So let's start with the introductory part. So basically the Ziehl-Neelsen stain, also called as Ziehl-Neelsen staining method, is a type of acid-fast staining method, and this method is mainly used to identify and to diagnose with the staining of those organisms like several types of bacterium and fungal species which have acid-fast physical property. This technique is also considered as a differential technique which is used to identify some acid-fast organisms which are mainly mycobacteria or several other types of bacterium. A bacterium which is a pathogen for the tuberculosis named as Mycobacterium tuberculosis was also diagnosed by the method of Ziehl-Neelsen staining. So before moving to the staining methods, let's understand first that what is the acid fasting and what are the acid fast organisms. So basically acid fastness is considered as a physical property of certain bacteria or sometimes of certain eukaryotic cells also, and in this condition these bacterium have the ability to resist the decolorization by acids during the staining procedures. So we know that what is the staining. Basically staining is the method or technique which is used to enhance the contrast in the samples. Different types of microorganisms like fungi, bacteria are identified or seen under the microscope with the help of staining by different types of staining methods. So for this there are different types of stains and dyes like methylene blue or many other dyes are used. So basically in the acid fastness there are several organisms which have the ability to decolorization, and that's why these organisms cannot stain by several other types of staining procedures. So basically for those organisms a specific type of staining method is used which is named as Ziehl-Neelsen staining method. So now let's understand about the discovery of Ziehl-Neelsen stain. So at first a scientist named as Paul Ehrlich developed a stain for the identification of Mycobacterium tuberculosis, and it is a species of Mycobacterium which is responsible to cause a disease named as tuberculosis in humans. So he named this as a hematoxylin stain, and after that a scientist named as Franz Ziehl altered this technique by the use of carbolic acid. So Franz Ziehl used this staining technique by the use of carbolic acid as a mordant, and after that a scientist named as Frederick Nielsen also modified this technique. He kept the Ziehl's choice of mordant, but he changed the primary stain of the carbol fusion, and on the names of their developers Franz Ziehl and Frederick Nielsen, this staining method is named as Ziehl-Neelsen staining technique. So this technique is named as Ziehl-Neelsen technique on the name of their discoverers. So now before moving to the procedure and method of the Ziehl-Neelsen staining, let's understand about the principle on which the method works. So basically the Ziehl-Neelsen staining uses a basic fuchsin and a phenyl compounds to stain the cell wall of Mycobacterium. Here the fuchsin is a type of red colored biological stain which is used as a stain or dye to staining methods, and this is also used to stain the cell wall of Mycobacterium species. As we have discussed earlier that the Ziehl-Neelsen staining method is mainly used for the Mycobacterium species because those species cannot bind readily to the simple staining methods, and that's why they use a heat along with the carbol fuchsin and phenol to the identification of these Mycobacterium so that they can stain properly. Here the use of carbol fuchsin uses due to it strongly binds to the negative compounds of the cell wall of the mycobacterium and also with the lipid cell wall of the mycobacterium species. So the mycobacterium consists of high lipid content in its cell wall, which is made up of mycolic acid, and this cell wall makes it waxy, hydrophobic and impermeable in nature, and that's why they cannot stain with a simple staining method, and they have the acid fastness ability. Now the addition of acid alcohol is also used in this method with the heat forms, so that a strong complex made that cannot be easily washed off. Here the acid fast bacilli take up the red color of the primary dye, which is used as carbylfuchsin, and other, which are non-acid fast bacteria, easily decolorize with the acid alcohol from the primary dye, and after that they take up the counter stain dye, which is used as a methylene blue, and they appears blue in color. So this is the working principle of the Ziehl-Neelsen staining, which uses carbylfuchsin as a primary dye and methylene blue as a counter stain dye for the diagnosis of different types of acid fast organisms. Now let's understand about the reagents which are used for the Ziehl-Neelsen staining. So as we have discussed earlier that carbylfuchsin, which is a type of dye, is used as a primary stain in the method of Ziehl-Neelsen staining, and acid alcohol is used to wash out, and methylene blue is used as the counter stain dye. So here the carbylfuchsin is made up of a basic fuchsin, which is a type of red dye used as a biological stain, and it is used also ethyl alcohol, phenyl crystals and distilled water. Now acid alcohol is made up of ethyl alcohol, which is diluted by distilled water and hydrochloric acid. The counter stain dye, which is used as a methylene blue, and 1% of acetic acid is made up of methylene blue, which is diluted by distilled water and acetic acid. So these are the reagents or stains which are used for the staining method in Ziehl-Neelsen staining. So now let's discuss about the steps and procedure of Ziehl-Neelsen staining. So basically this staining includes several steps. The first step is to clean the microscopic slide, which is a glass slide mainly, and then make a smear of the sample mixture or the sample culture in the glass slide, and after that heat fix the smear over the blue heat. Later it was stained with the carbylfuchsin, which is used as a primary stain in this staining method, and then heat gently until it produces fumes. And after that it is washed by the distilled water or tap water, by gently flowing tap water over the slide. Later it is stained with the counter stain, and the counter stain was completed by the methylene blue dye, which is made up of methylene blue and acetic acid. So flood the smear with the methylene blue dye so that it can be stained with the methylene blue as a counter stain, and after that leave it for 2 to 3 minutes and then wash it with water. Then it is air dried, and after that the observations were made under the microscope or under the oil immersion lens. So after the procedure completes, the slides were observed under the microscope, and here the acid fast bacilli take up the red color, and they observed red colored due to the carbylfuchsin, and those bacteria which are non-acid fast organisms easily decolorize with the primary stain, and then they take up the counter stain of methylene blue, and they appears blue in the microscope. So the results were made on the basis of the color obtained by these microorganisms. Now let's discuss about the applications or the uses of Ziehl-Neelsen staining. So basically the main important significance of this staining method is to identification of different types of mycobacterium species. As we have discussed that this is staining method is mainly used for the identification of Mycobacterium species, and the species of Mycobacterium named as Mycobacterium tuberculosis, which is a pathogen for the tuberculosis in humans, was first identified by this method also. The another application is this method is used to differentiate between acid-fast and non-acid-fast bacilli on the basis of the observations of color which are appeared in the microscope in these organisms. As we have discussed that acid-fast organisms take the red color due to the carbon dye, and those organisms which are non-acid-fast take the counter-stain dye and they appear in blue color. The another application of this method is they are used for the identification of some fungal species also, like Cryptosporidium and many other fungal species which can be identified by the method of Ziehl-Neelsen staining. Thank you.